presented by Caltech.
Hello, my name is Eric Winfrey. As you mentioned, I'm a professor of computer science, bioengineering, and computation and neural systems here at Caltech. It's a real pleasure to have the opportunity to introduce tonight's speaker and my good friend, Paul Rothemund. Paul is a research professor of bioengineering, computing and mathematical science, and computation and neural systems, a position he's held for the last 8 years. In fact, his connection to Caltech goes back a bit farther. He was an undergraduate student here, earning his bachelor's degree with a dual major in computer science and biology in 1994. That was when I first met Paul. I was a graduate student here myself when he was an undergraduate, and we took some classes together. Challenged by a computer science professor, that was when Paul designed his first molecular computer. I think it's fair to say that he was in no small part responsible for launching my own career, because I found his presentation on his molecular computer so inspiring that I ended up switching my thesis research from neural networks to DNA computing. Paul went on to do his PhD in computer science at USC with the father of DNA computing, Len Adelman. He was part of the team that, 1, built the most powerful DNA computer ever, and 2, determined that DNA computers were not likely to ever compete with electronic computers. So much for that. However, the mix of computer science and molecular engineering was a potent one that led to new questions in fundamental science, as well as many opportunities for creating new technologies. The bulk of Paul's PhD thesis considered how self-assembly processes can be treated as algorithms and studied with the tools of computer science. His work as a graduate student demonstrated remarkable depth as well as incredible breadth, from proof-laden theoretical research published in the top computer science conferences to painstaking experimental research published in the top science journals. After graduating in 2001, Paul joined my lab as a postdoc. I had just started as an assistant professor here the year before. Paul continued his research on self-assembly, including the seminal demonstration of what is now known as algorithmic crystal growth. However, the real magic happened when Paul disappeared from the lab for a while, and I didn't notice. It turns out he was busy inventing DNA origami, a technique for engineering molecular structures of incredible size and complexity. This work, published in Nature as a single author paper, rather rare thing these days, stimulated enormous interest in the field of DNA nanotechnology, and a whole generation of young researchers founded their careers on the possibilities that Paul exposed. Recognition of the importance of DNA origami didn't happen overnight. You might remember that the flyers for this Watson lecture show an atomic force microscope image of several smiley faces. Each of those things is a single molecule designed and synthesized by Paul. When Paul's paper first came out, that image made some people laugh. What good is a 100 nanometer DNA smiley face? They asked. At the time, it was just a demonstration of the level of control that Paul had established for molecular engineering. However, since then, DNA origami has found numerous applications, mostly in science, and papers using DNA origami now appear regularly in the world's top research journals. Paul has remained at the forefront of the field. His study of so-called stacking bonds established a new principle for hierarchical self-assembly that has allowed researchers to use DNA origami as a building block for even larger molecular assemblies. A few years ago, he pioneered RNA origami that can be synthesized by biological enzymes, setting the stage for applications of this technology inside living cells. This year's breakthrough is a demonstration that combining top-down lithographic fabrication with bottom-up DNA self-assembly makes it possible to build nanoscale instruments capable of measuring nanoscale photonic effects in one shot, measurements that previously required multiple painstaking, laborious experiments. You'll hear more about that soon. One thing that you'll discover during Paul's talk is that he is fearlessly interdisciplinary. His research delves into chemistry, into biology, into computer science, into physics, and melds them together. So you can't put him in a box. He's not a chemist, he's not a biologist, not a computer scientist, not a physicist. He is simply a scientist. It is a very Caltech way. Unsurprisingly, Paul is the recipient of numerous awards and honors. He was recognized with the Rosenberg Tulip Award in DNA computing, the top honor in that field. He received the World Technology Award in biotechnology, and the Foresight Institute Feynman Prize in nanotechnology. He was also named a MacArthur Foundation Fellow. That's the grant that's often called the Genius Grant. He's been invited to give TED Talks twice. Most recently, Paul was the recipient of the 2015 Essence Nanoscience Prize, the youngest to receive it. He has already had an outsized impact in research. All signs are that he is just getting started. Please join me in welcoming tonight's speaker, Paul Rothemund.
Thank you very much, Eric, for your kind introduction. Good evening, everyone. Thank you for coming. It's a great honor and privilege as a Caltech alum for me to join you here tonight. As the title of my talk would suggest, I'm going to talk about using DNA to build things, to build very small things. And the first thing I'm going to do is introduce you to my field. So it has over 1,000 researchers spread over 100 laboratories around the globe. And here at Caltech, including my own, we have 4 labs in this field of building things with DNA. And it makes Caltech a very wonderful place to pursue this kind of research. So if you want to look it up, the field is called DNA nanotechnology. Eric mentioned DNA computing. I'm not going to talk about that tonight. I'm just going to talk about building things. So what kind of things are we interested in building? So here's a catalog of molecular parts, the molecular machines that make up your cells and make your entire body work. There's a virus here to scale, just to show you what a cold virus is, but you can see the girders that give cells their form. You can see the motors that allow your muscles to move. If we zoom in, you can see the camera that makes your eye work. You can see the solar cells that harvest the energy for the biosphere. And you can see the switches that orchestrate the whole thing and govern biological development. Now, DNA nanotechnologists around the world are working to create all of these things out of DNA. But there's something I want you to notice, and that is that all of these things have very different shapes. And people say that form follows function. Well, there's a sense in which form precedes function, in that if you can't make a particular shape, you can't make something that you want. So shape is very important. Now, tonight I'm going to use a lot of metaphors. There's lots of different ways to build things out of DNA. There's something like DNA origami. There's something like DNA Lego. There's something like DNA carpentry. All of these are just many metaphors. Don't take them too seriously. They're a starting point for our conversation and for understanding. I also want you to think that everything tonight you see is artificial. We use DNA in a way that's different than the way biology does. And so if you think of us as carpenters, trees don't know anything about furniture. All right. Now, something I'm extraordinarily interested in is how we come to know the things we do, how we come to believe the things we want to believe. And if you're interested in that too, you could think of tonight's talk just as an elaborate way to convince you that DNA is in fact a double helix, right? The question is, why do you believe that this is the structure of DNA? We can go back to Watson and Crick in 1953. They got some data under highly ethically questionable circumstances from Rosalind Franklin. And she took this X-ray diffraction spot pattern of a crystalline fiber of DNA. And what Watson and Crick did is they interpreted that, right? So they used mathematics to interpret that and thought about it. They used some DNA model building, and they extracted the double helix. They learned that it's cleaved down the middle and has, these rungs that fit together like jigsaw puzzle pieces, and they extracted things like the twist of the DNA. That is to say, there are 10 rungs of the ladder for a single turn of DNA. Now, this business of determining structure with X-rays, X-ray crystallography, is a really important thing. It's been the gold standard for determining molecular structure for over 100 years. We wouldn't know how hemoglobin carries oxygen in your blood without it, but it's really very indirect, right? There's stuff missing from the spot pattern, and you can't take a picture. You can't take a portrait of an individual molecule. So it's very disappointing and frustrating. You have to sort of take someone else's word for the structure of DNA. Now, you could learn all these things. You could learn about the mathematics of Fourier transforms and do all this. But what if you wanted to convince a friend, right? You could take that hard way, or maybe you could take a different approach. Maybe a backward approach. Take seriously Feynman's quote, what I cannot create, I do not understand. And you could think up a DNA shape that was so extraordinary that it would be unmistakable, it would be big enough to see without X-ray crystallography, but it would depend so intimately on the structure of DNA that if you could build it, your friend would have to believe you. And what would be further great is if you could give your friend a method so that any time they wanted, they could build whatever shape they wanted out of DNA. They wouldn't have to rely on your shape, and they wouldn't have to think maybe you picked an easy shape, right? So DNA origami is just such a method. And tonight, what I'd like to convince you is that it both has interesting potential applications and that it's a very beautiful way to sort of demonstrate the structure of DNA. Okay, so we have to start with the premier DNA carpenter, Ned Seaman at New York University. And so what happens is he was a frustrated crystallographer like Rosalind Franklin, and what he wanted to do was, what he finally figured out, because he couldn't crystallize proteins very well, is he wanted to build DNA cages that were so rigid and so good that they would hold their protein prisoners still enough that you could take an X-ray mugshot, right? That's what he wanted to do. That was his research program, and he's still doing it 35 years on. But along the way, he had to create DNA nanotechnology, right? Technology, this business of building geometric forms of DNA. And if we zoom in on one of his first ones made in 1990, you can see that one of the major contributions is that DNA just doesn't have to be a double helix. It can have joints, right? So it can have a place where three sort of cylinders, three rigid double helices of DNA come together, and a DNA strand can wind along one helix and then jump to another helix, right? So this is DNA carpentry. I won't go into it. But back then, it was still pretty hard, right? So it took Ned a year to design, to synthesize, and prove to himself and everyone else that this was, in fact, something that he made out of DNA. Part of it was it was so small, it's just 7 nanometers. But part of it is that there were some fundamentally laborious parts to sort of this DNA carpentry approach. And so what happens is that actually a Caltech alum, after seeing a talk of mine in 2004, Jonathan Kelly, asked me if there might not be an easier way. Would there be a way for us to, without spending a year thinking really hard, without us being Ned, the expert, could we come up with a way to make any shape or pattern? And my answer was to turn to origami, right? So somehow come up with a way of taking a long single strand of DNA and folding it into any shape or pattern which you might want. So the question is, what DNA to use as paper? And so it's hard, for reasons in principle, to get long single strands of DNA. And so you can look to a natural source. And if you look inside of this human, you'll find a 3 billion base paired genome. But it is maintained as a double strand, so that's no good. So if you look deeper inside this human, you'll find the bacteria in his gut. And they maintain their genome as a 4.6 million long strand of double-stranded DNA, and so that's no good. We're still looking for this single strand. But if you look inside of them, you'll find that there's viruses that infect only bacteria, and they maintain their genomes as single strand. And here's one called M13. It was discovered in a sewer in Munich, Germany. And because of its appeal to the biologists, we now make 1 billion times more copies of this virus for biotechnology for evolving proteins than the total number of chickens and cows on the planet. So this is a very much bred DNA sequence. And you can buy it cheaply from Bayou Biolabs in Louisiana. So now, so the question is, if we have this long strand, how are we going to fold it, right? So this is just a small section. It's about 70 bases. It's about 70 of the DNA letters of that 7,000 long sequence from the virus. And what we're going to do is we're going to break the origami metaphor. We're going to add short strands, which we call staples. We're going to add about 200 of them. And each one of the staples has the following form. It has a left half, and it has a right half. And the left half binds in an ATCG pairwise way, the famous Watson-Crick binding. It binds one position on this long strand. And in the same Watson-Crick pairwise way, A pairing to T, C pairing to G, it binds a distant location. And it brings those two distant locations together. It enforces a constraint, and that's our crease. And the net action of 200 of these things, making all kinds of creases, fold this long strand into some shape, for example, a rectangle. Now, I'm going to show you a movie. Here's a long strand, and here's a short strand. This is just to give you an idea of how this process unfolds in a test tube. It's a spontaneous process. This movie is a cartoon. It doesn't really reflect really what's going on. Here, the DNA strands whiz in from off stage and magically go to where they're supposed to go. That's not what it's like at all. In fact, the strands are bouncing into each other. Staples are binding the long strand and unbinding as a function of the temperature. At high temperature, all of the structure is melted away and they're not binding. At low, as they cool, longer and longer pieces start to settle into place. And if you don't do this process of cooling at a defined rate, then the thing will just become a bunch of tangled glop. Okay? So now when you see a finished DNA origami like this, you can see that what it really is, it looks a lot like regular DNA. It has a bunch of parallel double helices. And then it has a bunch of crossovers. These crossovers are what hold the double strands together. And so there's a staple that goes like this. It goes along one strand, and then it jumps to the other strand and goes back again and enforces a constraint and holds these helices together. So this is the basic structure of the first version of DNA origami. So now, to sort of create a structure that couldn't be mistaken for anything else, a structure that would show that we could actually fold DNA into complicated form, I designed this fold for the long strand. So I'm not showing the staples, I'm just showing this fold for the long strand. And after you fold it, you still have a little bit of DNA left, so you leave a little loop at the bottom, right? So now, let's get an idea of how big this is. If we zoom out, we can start to see this very large curvature here, right? This is the curvature of a red blood cell. So if we zoom out, you can see that 70 of these fit across the width of a red blood cell. And so it's extraordinarily small. And so we can't look to normal light microscopy to see them. So we have to look to atomic force microscopy, a method for measuring molecules that was invented in the 80s. And the first thing we have to do is immobilize our molecules on a hard substrate. So we stick them down very firmly, and so these sort of rectangular things are surrogates for molecules. And then you take a very fine needle, a micromachine needle, very much like the needle in a record player, and we drag it back and forth over the surface. And it bumps up and down, and we shine a laser beam at the tip of the arm that holds the needle, and we measure the deflection of the needle as it goes up and down. And when it goes up really high, we record a white pixel, and when it goes down low, we record a black pixel. And in that way, in a computer, we build up an image of something that's very much smaller than you can see in a light microscope. So how do we go about, just a little more details of how do we build these things. We design them in a computer program. It spits out a list. We send the email to a company in Iowa. They have a robotic chemist called a DNA synthesizer. It builds up these 200 strands, AC, GT, in an automated fashion, and they send back in FedEx 200 strands. Right? So, and then you can mix them all together. You could mix them together in your kitchen. You heat them up to near boiling, and you cool them down. And what you get is a tube of 50 million. You get 50 million DNA origami in a single tube. Now, remember I said in order to do this AFM, this atomic force microscopy, we have to immobilize them very well. So what we do is we harvest mica from the ground. It's a mineral that comes from granite. You dig it out of the earth. You peel off a layer with Scotch tape, and it's extraordinarily flat. And it's sticky for the DNA. And so the DNA stick extraordinarily strongly. And then you're in a position to take that very fine tip, that tip that's about as sharp as a DNA helix, and drag it across, and then take a picture of actual molecules. All right, so you can see a bunch of DNA origami, and you can see that they're not all perfect. Some of them have their jaws flipped over their head. Some of them have their noses punched out. But the basic idea is that it worked, right? You can zoom in and you can see that the fine structure of the thing that you wanted to make, even down to the loop hanging off the bottom, the little goatee, it's all there. Now, if you design structures that are a little more rigid, then you can get a higher yield. So these are some rigid triangles, and you get greater than 95% of these. So over the years, very quickly, people extended DNA. So now the question is, how well are we doing, right? So we decided we wanted to make molecular forms, and if we add a DNA origami, this is a DNA origami compared to all these molecular machines. You can see here's Ned's cube, and you can zoom in, right? And you can see that it's the case that while we have managed to make a very high resolution structure, we haven't been able to make structures at the same resolution as the protein machines. So we sort of have this kind of few nanometer resolution for making a smiley face. Here's this cube. And so a way to think about it is that we've managed to make any arbitrary shape, but we haven't quite reached the limit that nature has. So if you think about it, There's sort of kids Lego, and there's sort of adult Lego. And DNA is kids Lego, and protein is adults Lego. And there are a large number of people who are trying to figure out how to take the next step and push it down so we can make whatever shape we want at that level. Now, I just showed you 2D origami. And very quickly, there are a whole bunch of people who are making 3-dimensional origami. So there's no obstacle to folding the long strand into a 3-dimensional form. And so William Xi's lab immediately made a very large number of interesting 3-dimensional structures. So that's the fast and easy extension. What was a surprise to me is that Hendrik Dietz, who's in William's lab, thought about using DNA appropriately as little springs so that you could add bases or subtract them and add torques to the structure so that you could get twist and curvature, and you could make a variety of elaborately curved forms. And another lab, Jan's lab, has done it in a slightly different way, and they've made beautiful spheres and beautiful, it looks like a sort of a clay coil vase or bottle, right? So they've made these. And one of the things that's neatest to me lately in Jan's lab is that they've abandoned the idea of holding together these parallel double helices all together and abandoned the sort of stiffness that comes from them and made these sort of wireframe structures, right? And so if ever there was a structure that tested the idea that we have a good idea of how to make a curve or a bend in DNA, this is one of them. And I'm going to flash the atomic force micrograph, you can see that they succeeded in making this very unusual shape. And if you look at, so that's just one of them, if you look at a different one, you can see that something's funny in the flower, right? Its stem may be a little bit weak. Sometimes it bends over and flips around, right? So we can learn something. Rather than using X-ray crystallography, we can really learn something deep about what these molecules are actually like, their properties. So that gets us back to these smiley faces and what was wrong with them. And so William Xi at one point, very soon after this, came to me and said, dude, your origami are twisted. And in fact, it's the case that I had gotten the DNA origami wrong. Right? So I had used the wrong twist. So remember, Watson and Crick had figured that there were 10 rungs to a turn, right? So I had used 10.67 bases per turn, rungs per one turn of the ladder. And I had done it for what I thought were some good reasons. I thought I could get away with it. If you take a very long chain of DNA origami and you deposit it on mica, then you see that it fragments in this very funny pattern. So the DNA rectangle chain is very long, and it's very sticky for the mica. So if it's twisted, it goes down and breaks. And if you use the right twist, 10.44 or thereabouts bases per turn, it becomes stick straight, right? And you realize that you made this mistake. So if you look at X-ray crystallography, out of X-ray crystallography, you get 10 bases per turn. Well, much later X-ray crystallographers all got 10.4 or 10.5 bases per turn. But the point is this, is that if you are building something from DNA, there's no room for argument. You can't wonder about whether one experimenter did it under one conditions or another. If you're actually building these structures, it's very critical that you get it right because you can't build what you want. So it really shows you that you understand the structure of DNA. In the time since, there have been a very large number of different machines and devices made out of DNA. And I'm sad that I can't talk about all of them. There are little calipers that can measure a distance smaller than an atom. There's little syringes that can grab onto something that looks like a cell and insert this end and make a little syringe hole through a cell. So there are all these amazing machines. Now I'm just going to talk about one today. So since about 1966, there's been this fantasy, at least since 1966. You all potentially remember Fantastic Voyage with Raquel Welch. The idea that you're going to inject robots, or actually, and they were injecting submarines, but you're going to inject some kind of miniature device into the bloodstream. And why would you want to do this, right? So here's one of these devices. Potentially, it's turning on a light for some reason and has a camera within your blood, right? But the point is, what's the power of this? The power of this is that you're adding some kind of recognition, some kind of ability to make decisions in a drug that we do not have. Our drugs are too stupid. They can't make decisions. And so, for example, we employ a chemotherapy. It goes into your body, and it's trying to distinguish between cells that you want to keep and the cancer cells. And the best differentiation we have often are between cells that are growing fast and those that are not growing fast. And so when we give these kinds of drugs, your hair falls out and you get otherwise sick. So the whole reason for adding a computer chip to this thing, right, is that you want to be able to make decisions about what you want to attack or what you want to do inside a body. So the reality is much more mundane than that. So this device is not something that was done in my lab. It was done by Sean Douglas, Ido Bachelet, and George Church's lab when they were at Harvard. And it really approximates these styrofoam clamshell sort of takeout container. And what it does is it encapsulates the drug of interest. And the idea is it's supposed to open if and only if it touches a cell that's the one that you want to deliver the drug to. And indeed, it really looks like this. So this is a 35 nanometer picture of it, and there's a sequestered antibody payload. That's the drug, okay? And so if you look at a 3D representation of it, you see that it actually is open on the ends. So it's not a very sealed container, but these molecules are big, and they don't really wander outside of this. So it's okay that it's leaky on both ends, right? And if you look at one of these closed clamshells, you can see that it's a compact form and has the payload in it, either gold molecules or little proteins. And so you think you can make a closed clamshell. Now, the real question is how you actuate it, right? So it has an opening mechanism. It has DNA locks on it. So these are very special DNA locks. And if you zoom in on those, you'll see that they have a funny form where the DNA double helix is stable until you present it with some kind of ligand. You present it with something that you're trying to recognize, at which point that thing that you're trying to recognize pries off one of the strands and releases the two double strands. So you can see this is a method for opening the clamshell. There's a ligand that's bound to one of the locks, and there's another ligand that's bound to one of the locks, and they've opened it, and they've exposed the drug inside, right? So if you look at this in 3D, this is rendered as if it's really sprung open, but in fact, the molecule will be fluctuating. But if you take an electron micrograph of it, you can see that a good fraction of them are actually open when you add the keys to the locks. All right. So what's the picture for how this is going to work? You have your DNA nanorobots, and you have two cells that you're trying to distinguish, good ones and the ones that are supposed to be cancerous. And there's the drug, and the drug in principle would affect both of them. There's a place where it could affect both of them. So it can bind to both of them, and these particular things that it can bind to are surrogates for things that might have some immune action. They might recruit other cells to kill these cells, right? So you have a drug that you want to only apply to this red NKL cell. So there are the locks, and the key to the locks occurs only on the cell that you want to interact with, right? So this is the setup for being able to distinguish between these two cells, right? And the thing that you want to have happen is you want this cell to be untouched, and you want the clamshell to recognize this one, the key to open it, and then, right, it opens and does something. So the picture is that if you have one of the, so we'll just go through the logic of this. In this picture, you have the cells that you don't want to kill, the cells that you do want to kill. You have whether or not this is the case that they have not in fact been bound by the clamshell. This is the case they have. So if you make a clamshell with locks that don't open and you apply it to both cells, it's good because both of them don't bind the clamshell. If you make a clamshell that's open, right, it's just open, then it'll stick to both of them. So in fact, both of them were able to bind the drug. But if you use one that has the locks actually functional, then only the ones with the keys shift in this assay and show that you've in fact bound and opened the device on the cells that you wanted to affect, right? So the question is, in what sense can we dignify this as a nanorobot? And the answer is that it can sense things, right? And it can act, something we'd like to do. So cancer is very complex, and it's not clear. Cancer is not as simple as finding just one kind of key. Or even just recognizing a particular tissue in your body is not as simple as finding one kind of key. So what's been added to this is the idea that you can have multiple kinds of locks, right? And so now, and multiple kinds of keys. So now, Sean Douglas and Ido have shown that you can take these devices and open them if and only if there are two kinds of locks. And this has allowed them to distinguish many different kinds of cell types. So now, you can ask, there are many difficulties with this, and you might get excited. There's sort of the good news. This is the good news. We can recognize this in different kinds of cells. But there's the bad news, right? This is extraordinarily complicated. Could this ever work in a human? And William Shih has explored this. So this is a highly complex drug, right? So if we zoom in, right, penicillin has just 41 atoms. This has hundreds of thousands of atoms. So William Shih has explored this in a living animal model, and you can see that it might act differently than something like penicillin. And what happens is this. So this is not the DNA origami that we've been talking about. It's not the clamshell. But if you introduce it to a mouse, you'll find that within 30 minutes, it breaks down and/or finds its way into the bladder, right? And the mouse sort of pees it out. So the good news is, right, so that's bad news. So the good news is you can encapsulate it in a lipid layer. Now it's greasy. And 2 hours later, it's still circulating in the blood of the mouse. So that's great. So this is a neat advancement. But then it's the case that what if you tried to use those clamshell nanorobots? Well, presumably they would work the same way. This one would end up where it does, and this one wouldn't. But now it's the case that you can't open the clamshell, right? So this is a way to do this, but you sort of defeated yourself. So that's bad news. So the point is, on this story, it's extraordinarily complex and it's long-term and sort of have to stay tuned. William Shih is sort of already kind of making solutions to this. But the point is, this is a long road, and we'll see where it turns out. But it's one that's very exciting, right? OK. So now let's consider a different fantasy. So here's another fantasy. This other fantasy is that you think that you're going to somehow self-assemble computer chips or cell phones. In this autonomous way, you're going to mix up some DNA strands together with some molecules. And because the molecules are smaller than the transistors in these computer chips, that somehow you're going to be able to make a circuit with hands off, and maybe cheaper, and maybe more energy efficient, all these things. This is a fantasy that people entertain. So how might we go along the route to this fantasy with DNA origami? So DNA origami has the following property. The staples each find a unique place in the final origami. So the green one goes right there, and the orange one goes right there. And this process allows you to make patterns. So for example, you can attach devices, here little bumps, onto different staples, and they will go to where you want, right? So you can program the pattern that you're making. You can organize things by attaching them to the staples and then letting the thing assemble, right? So here's a picture, right? So these are a bunch of different patterns that you can make. So you can easily make very high resolution patterns this way. And so then the cartoon becomes, we're going to attach either you were going to attach devices to the staples, and then we're going to let the thing assemble, and then maybe we have some kind of working circuit, and then we could even, if we needed to, wash the DNA away afterwards. But there's something missing from this picture, and so I'm going to go through an example of where we tried to do something like this. I hope you can detect what's missing, and then we'll talk about that. So here's one of these amazing molecular devices. To scale next to the cube, it's a carbon nanotube, and it has amazing conductive properties, and it has some interesting properties that make it good potentially for electronic devices. So one property is they come in two flavors. They can be metallic, like copper wire. They can be semiconducting, like silicon. And if you manage to make a cross out of a metallic one and a semiconducting one, then just having done that, you've made a transistor, right? So you've made something called a field effect transistor. And that means that if you put a battery across the blue one, the semiconducting one, a large current may pass. And then if you add a battery, you start adding a battery to the red one, and you turn it on, it will turn off the blue one. So now you've created a valve. You can increase the voltage on the red one and decrease the current on the blue one, right? So this is a simple transistor. And the way people originally tried to make them was a kind of a simple way. You would play pickup sticks with the carbon nanotubes, you throw them on the floor, and then you'd use atomic force microscopy or something special to find the crosses, and then having found them in this laborious way, you'd wire them up, and voila, you have some devices. And this is not a potentially good way to get yourself to a scalable kind of technology. So a bunch of students at Caltech decided with us, and a bunch of professors decided that they could potentially do better with DNA origami, right? We're going to make these crosses. And so you start with an origami with DNA strands on it hanging off like this. You start with blue and red carbon nanotubes that have appropriate DNA sequences on them. You mix them up. So far, so good. You're going to make some crosses. You take some atomic force microscopy. Maybe the crosses don't exactly overlap the way you want, but some of them do, so you can make a lot of those. You put them on a surface. They're the little tiny carbon nanotubes, giant gold electrodes. You wire them up, and then you can measure them, and you can measure that if you just measure the current on the blue tube, you can get a high current, and that as you start adding batteries to the red tube, you increase the voltage on it, the current on the blue tube drops. Congratulations, you've made a field effect transistor. This is all very exciting, but I have not given you the complication that how did we find these? Well, how did we make these? Well, you organize the crosses in solution, because that's where DNA origami starts out, so you make the crosses there. And then they act as then they're like a bunch of playing cards. You throw them on the floor and they go all over the place, and you have to find them with some kind of fancy microscopy, right? And then you have to wire them up. You have to make these idiosyncratic electrodes that sort of follow these weird cow paths all the way from the macro scale with your hands all the way down to the DNA origami that you can't see there, right? So now all of a sudden you've made, maybe potentially, if you did well, you've made all these crosses, all these devices, and you can't do any better than the guys playing molecular pickup sticks, right? So this is what I might call hunt and peck connect. It's very hard to do, and you're not, again, going to have a scalable architecture this way. So with scientists at IBM Research, so let me just back up. So what are we trying to do here? The important thing is we're trying to be able to integrate things together. So the success of your cell phone, right, is that we can integrate different kinds of things on the same computer chip. Here's an electronic computer chip. Here's a mechanical chip in the middle. This is an accelerometer, like the ones that set off your airbags, and it's like the ones that tell you your cell phone the orientation that it's in, right? So the ability to integrate these two different things together has given you enormous power. Now your cell phone knows where it is in space. So what we want to do is we want to be able to integrate molecules with this. If the molecules are so great, we want to be able to put a carbon nanotube right there, or we want to be able to put a bunch of them in different places. So the goal is to find some way to marry this very successful microchip fabrication and the molecules. And with IBM Research Almaden, we addressed this question. So I thought it would take a long time, and within a couple years, they came up with a very simple idea that all you had to do was make binding sites for the DNA origami. Right? So you would use the same technology that you use to print the computer chip to make sticky patches in the size of the shape in DNA origami. If you added those very rigid triangles and maybe a little bit of glue, some positively charged glue that glues the negatively charged DNA origami to the negatively charged sticky patches, you could organize the triangles and they would all go exactly where you want them. So you pour them across the chip and they land and stick exactly where you want. And the idea here, it's kind of paradoxical. The idea is before we were using DNA origami because it had these small, fine features to organize the parts. Here, we're using its largest feature, its outline. Because DNA origami are giant molecules, they're just big enough that we can begin to use our finger and write sticky patches that allow us to bridge the length scales, bridge the sort of microchip length scale and the molecular length scale. So this is one of the things I'd like you to take away, that these molecules are so big that we can actually make a molecular binding site sort of by drawing it with our hand. Okay, now many problems remained. And so, for example, the magnesium would cause carbon nanotubes to clot and everything else you want to do. So Ashwin Gopinath, a postdoc with me, he spent 4 years reimagining and refactoring and completely changing this process to make it extraordinarily good. And it's the case that I am going to gloss over all of his work and merely admire the result. All right, so we're going to merely admire the result and see that we went from a situation where all the triangles are on a surface like this to they're all organized on a surface like this. Right? And Ashwin would want me to tell you, that greater than 95% of the sites have triangles and they're well aligned, and this works on any semiconductor surface you might want to use. So that's great. And we go back to this situation to say, okay, have we solved our problem? Right? And so potentially you could attach these carbon nanotube crosses, and that would be great, and then you might be able to wire them up, but the astute among you will notice that these triangles can't get you there, right? Not equilateral triangles, because they'll go down in different orientations. So this is not a way to do this, and rest assured, we've actually solved this problem. We know how to use asymmetric shapes that aren't equilateral triangles and get these to orient the way we want. Nevertheless, I'm not going to talk about pursuing this, because there are lots of other problems with carbon nanotubes. They're hard to solder to, for example. That doesn't work so well. So we're going to talk about going in a different direction. So with a team of Ashwin and Evan Miyazono and Andre Ferron at Caltech, we decided to explore physics applications and optical applications that did not require soldering to molecules. And we're going to start with a pinball machine, right? So this is a pinball machine. It should be familiar to all of you. This is a quantum pinball machine, right? Otherwise known as a particular kind of quantum computer. And the way it's a pinball machine is the following. On this end, you're going to put in pinballs, that is light, photons. And at the other end, you've got the scoreboard, which are a bunch of light bulbs that are going to light up. And what happens, let's focus on a couple of these pinballs. They enter the pinball machine, and then they reach a junction when they encounter each other. And then they come out of the junction, and if there are two of them, and they're perfectly the same color and everything else, only one emerges. So it's a funny pinball machine. That's what makes this a quantum pinball machine, right? Two pinballs end up as one, and then it goes to the end, and it lights up. And what makes this a quantum computer is that it's actually difficult, based on the set of input pinballs, to figure out what pattern of lights light up at the end. And really what you do is you flow through millions and millions upon millions of pinballs, and you measure these light bulbs millions upon millions of times, and you build up statistics, and you measure correlations between these light bulbs, and that's how you figure out that it's a quantum computer, right? So this is probably one of the most complex quantum computers that's ever made. It was made in 2014. It's got a lot of parts to it, right? So here's the pinball part. That's the pinball part. This is the computer that controls the flippers that are at these junctions in the pinball machine. Here is the source of the pinballs. It's a giant laser, and there's a crystal that makes sure that only single photons come out of it, right? So this is a very complex piece of equipment. And then there's the scoreboard on the other side. And we're going to concentrate on this part, right? We're going to concentrate on the light source. And the reason we're going to concentrate on it, so again, remember, we're trying to integrate everything. The thing that's enabled the modern world is our ability to mix electronics and mix optics and mix micromechanics all on single chips and put them on our cell phone. So what we're going to do is this part right here, this part of the pinball machine, already occurs on a chip. And there are other people who have done a really good job of putting these kinds of things, the scoreboard on chip. And so we want to put this thing on chip. So this thing, which has the giant laser and the crystal and everything, can occupy an optical bench the size of a pool table, and it's extraordinarily expensive, hundreds of thousands of dollars. So the question is, how can we do this with something that's more of a computer-chipped kind of technology? And the answer is, we're going to zoom into something you can fabricate with computer chip technology, right? So what is it? So it's just you can think of it as a thin glass membrane, and that thin glass membrane has a bunch of holes punched in it, right? So there are all these holes punched in that membrane, and at the center of that suspended membrane, there's a little area, and that little area is like a drum for light. It can resonate with light of a particular frequency. So what do we know about drums? So here's a drum. We know that drums have different so-called resonant modes. They can oscillate in different patterns. They can bounce up and down like this, or they can bounce up and down in a somewhat more complicated pattern. So if this is a drum for light, we're just going to look at the central area. That's sort of the head of the drum. Then we know that there can be and there is, for a particular wavelength of red light, a very complicated pattern of oscillations of the electronic field there, right? So we know that this thing has a complex so-called resonant mode, right? And we know so now we think about a drum, right? It's a resonator. It doesn't actually make any sound unless you hammer on it. And just like a drum that you have to hammer on, you have to hammer on this little drum for light to get any light out of it, to cause it to dribble out photons into our pinball machine, right? So we're going to add light bulbs to this. And it's important that we get the light bulb in the right position. If we get it in the center in one of these bright spots in the resonant mode, that's good. It'll be bright. If we get it in the wrong place where it's dark, it'll be the light bulb will be dark, and this little thing will not produce anywhere near as much light. It could be many-fold, or it could be, in some cases, hundreds, okay? And it's the case that, not for all applications, but for in particular some quantum computers, it's important you don't get two in there, right? You don't want to have two light bulbs in there either. So how would people do this in sort of the different old paradigms? So one way is you start with a blank piece of a surface, and you actually build the resonators ahead of time, right? So you're hopeful, and you build all the resonators, and you build some little optical wires to them so light can leak out of the resonator along the wire into your pinball machine. That's great. And then you throw light bulbs down all over the thing and hope that one lands exactly where you want in one of the resonators, and that they don't get a dud, it's in the wrong place, or that two of them don't land in the right place. So this is the spray and pray approach, right? So then there's another approach, right? Equally, you start with a blank surface, and you say, okay, I'm going to put a bunch of light bulbs down. And this is similar to the other approach with the carbon nanotubes. Now you're going to look for ones that are sufficiently well separated, they're only single ones, and that you can draw the wires to. And so now you can make a resonator here, and you can make a resonator here. So this is sort of the Hunt and Peck Kinect approach, right? So these are the previous kinds of approaches. So we want to do something different. So we're going to use DNA origami, and we're going to put the light bulb on the DNA origami, right? That's sort of obvious that that's how we're going to start. And the light bulb is not a light bulb. It's actually a little organic molecule. And they're actually very much smaller than a light bulb. And in this case, we actually, for the purposes of this experiment, we put a bunch of them on. So from now on, if I draw a white triangle like this, just know that it's got some appropriate light bulbs on it for the experiment. All right? So we know that with our technology worked out by Ashwin and by IBM Research, that we can park a DNA origami with these light bulbs anywhere we want within this cavity. So in principle, the problem should be solved. But how are you really so sure that you're actually going to get the origami and the light bulb in here? How are you sure that this is really the resonant pattern in the first place? Maybe you didn't get things quite right. How do you really know what's going on? Well, you'd like to be able to just look, right? You could use a light microscope, and maybe you could see that resonant pattern inside the little photonic crystal cavity. But you can't, right? And that's because that cavity is basically the size of an E. coli. So it's actually below the diffraction limit. It's smaller than what you can see with a light microscope. And it's the case that all you would see is a big blur, right? So in a light microscope, this is what you see. So you're going to have to figure out how to do it some other way, right? So inspired by Doug Smith, who's a science writer at Caltech, he helped me think about that we might talk about microwave ovens. So it's a kind of similar thing. It's a resonator. There are microwaves that are bouncing around in here. If you're like me, you have an old microwave that doesn't have a turntable in it, and there's a problem. You know, you can try putting your soup in the center, but that's maybe not where the best place in the resonant cavity is. Maybe it's not going to get hot there. So maybe it might get hot in this corner. It might get hot in another corner, right? So you've got to figure out, you know, where the hot spot in your microwave is to get your soup going. Okay. So as any Caltech undergrad knows, the answer to this kind of thing is PEEPs, right? So if you put the PEEP in the microwave in a cold spot, it's going to stay the same size. If you put it in the microwave in a hot spot, it's going to blow up, right? So then the picture is you're going to put a bunch of PEEPs in your microwave like this. You're going to turn it on. Some of them are going to get big, and you're going to, aha, I can put my soup here or here, okay? So that's really great, okay? So that's a good idea. But if you think about it, it requires you to stand up pretty close to this microwave. You have to be able to peer inside. And that's like the situation with the light microscope. It's really the case that this would all just sort of, if we were transferring this to the realm of the little nanophotonic optical resonator, we would just see this kind of thing, all right? So you have to keep on thinking about microwave ovens for a minute and try. So you're going to try 2.0. Your friend's got another microwave. You're going to try it again. And this time you run out of PEEPs, so you're going to use firecrackers, right? So this is really good. So now you're really smart. You're going to do the same thing you did with the PEEPs. You put a bunch of firecrackers, you turn on the microwave, and then it goes like this, right? And so there went your picture. Again, you don't have access to this. So you have to think a little bit harder. And your friend says, well, I got a bunch of microwaves, right? And you're like, aha, if I have a bunch of microwaves, I can do something, right? So you're going to put them in a giant grid, right? You're going to put them on a giant grid, and you're going to put a firecracker in each one of them, right? And you're going to do it in the following way. So you're doing this out on the football field, a safe distance away from all these microwaves and firecrackers. And so now if you look in the upper left-hand microwave and you zoom in, you see that the microwave is in the back left corner. And if you zoom in on the one in the middle, you find the microwave is in the middle. And if you go to the lower right-hand corner, you find the microwave is in that position. So there's a correspondence between the position of all the firecrackers and the position in this much larger array of microwaves. You can stand away at a safe distance, 1,000 feet away. You can see it with the naked eye, right? And now you let it go, and you have a pattern, right? You know, a bunch of the microwaves catch fire. You go, aha, this is the pattern for this model of microwave. If they're all the same, this is the pattern of hot and cold spots in the microwave. So we're going to do the exact same thing with these little photonic crystal cavities, right? And because we can make them on a computer chip, we can make a lot of them, right? We can make hundreds and hundreds of them. And in the exact same way, we're going to make a grid of them. And in the upper left-hand corner, we're going to have one. And we're going to put the origami with the light bulbs in the upper left-hand corner. And in the one in the middle, we'll put the origami in the middle. And in the one in the lower right-hand corner, we'll put it here, right? And then we're in the situation that we made this giant array, something that we can look at with an optical microscope. And when we take the picture, we see a pattern of spots like that. So now we can actually visualize the resonant mode of this little drum for light. And it's the case that it matches very well with that's theoretically predicted. And what you've done is you've sort of enacted a 200-fold increase. You've gone from something the size of bacterium to a microscope that can see, you know, a human hair, right? So these are sort of the sizes that you've managed to accomplish, right? So now, really the thing that you're interested in is control. And so here we made a measurement, and we showed that we could measure this pattern. But what it really shows is not so much that you can measure the pattern, that we were right, that we could put the light bulb exactly where we wanted. And we wanted to show just a little bit, a different level of control. Not greater, but just a different level of control. It's not good for the quantum computers, but sometimes you might want to be able to control the intensity of this thing in a digital way. So we could put one DNA origami in the middle of this thing, and then add another one at a different peak in the mode, or add another one. And so now you have sort of digital control over the intensity of this resonator. Right? So now these things can become pixels, right? Resonators that have 7 of these have one intensity and none have another. And you could now think of them as pixels, and you can zoom out, and you can make a pattern like this. And in case you can't see it, it's that. Right? So what we've shown here is that we can do this at very large scale. That we can make 65,000 of these little drums for light, and we can basically get them, almost all of them, to do what it is we want by installing molecules via DNA origami. All right. So the question is, can we really replace the big optical table, the pool table that feeds into this quantum computer? And the answer is maybe. There are lots of things wrong with our light bulbs. They blow out very easily. Their color is too muddy. They're not a perfect pure red color. They've got a little bit of orange and a little bit of a different red. And so there are lots of problems with them. We're working on better light bulbs, right? So there are lots of different light bulbs you can try. But even if this never works for this kind of quantum computer, and it may be that today this kind of quantum computer may even be passé, people may be on to other kinds of quantum computers, the point is that our ability to install these kinds of light sources into microfabricated optical devices could enable all kinds of things. And people have suggested to me, atomic clocks on chips. There's all kinds of telecommunications you might do, and there's a lot of fundamental physics questions that you can ask if you can install different kinds of optical devices that you can't make in silicon in the optical devices. So that's one answer. Another answer is that there are other interesting things to do. So this represents, I don't want you to look too deeply at this, but this is just a method for DNA sequencing, and one of the most amazing methods worked out by Pacific Biosciences. And they use, here's a grid of little holes in a surface, and they're not exactly like those resonators we talked about, but in some ways they're similar. They amplify and confine the light from single molecules sitting at their bottom. And so the kind of technology where you can use DNA origami to integrate and install single molecules at the bottom of such wells may be able to do things like help you with DNA sequencing or other kinds of molecular detection. So there are a lot of potential applications for this technology. So finally, I'd like to review. And I'd like to remind you that by building things, right, you can have a more direct experience of something, a more direct experience of the structure of DNA. And the more complex a thing you build, the fancier thing you build, the more certain you are that you understand the structure of DNA, right? Because it relies on finer and finer features, right? So that's a very interesting thing about how we come to know things. I'd like to remind you that we can make any DNA shape or pattern that we might want. Really, I hope I convinced you of that. We can't make things down at the level of protein yet, right? And so we and others can be working on scaling that down. But really, we can make any shape we want. Our ability to make things at that length scale, to make any shape we want, allows us to make the most complex medical devices that I think we've ever made, potentially. So the most complex, we certainly can make the most complex molecular devices, but these may be, they're complex enough that they could have bearing on some very interesting medical challenges. And then I'd like to remind you of this funny paradoxical thing that the fact that a DNA origami is so large allows us to begin to integrate it with the most successful technology we already have, that of computer chips. And we can do it at a large scale. We can make tens of thousands, or in principle, millions of devices. And we can really be able to take the power of molecules. They have interesting optical, electronic, biological properties, and integrate them with silicon. That's it. Thank you very much.